Hello, my name is Ryan Barnes. I'm a postdoc in the lab of Patty Apresco at the University of Pittsburgh, and my poster is entitled Telomeric Adeoxoguanine Drives Senescence Independent of Telomere Shortening. So, telomeric adeoxoguanine is a base damage that occurs at the telomeres, which are TTA-GGG repeats in humans, and can arise under conditions of oxidative stress, or when the level of reactive oxygen species outweighs the level of antioxidants within a cell. Our lab is particularly interested in telomeres because they are sensitive to oxidative stress, and correlative studies have shown that telomere shortening is accelerated in conditions of oxidative stress, and this is believed to enhance aging. However, there has been no way to specifically target adeoxoguanine to the telomere until recently when our lab has published on a tool which uses the FAP technology, which is a protein that can bind malachite green derivative dyes, and when exposed to far red light, produce singlet oxygen. So our lab fused the FAP to TRF1, a telomere binding protein, so we could specifically induce singlet oxygen-mediated adeoxoguanine at the telomeres. So I introduced the FAP technology into two normal, non-diseased human cell lines, BJ and RPE cells, and you can see here the colocalization of the FAP with the telomere red PNA fish. And since these are non-disease cells that have intact signaling pathways, we assumed that we would induce senescence, and we did. Only 4 days after treatment with dye and light one time, we saw phenotypes consistent with senescence, including beta-galactosidase activity, laminin B1 loss, and the SASP. And we also confirmed that telomeric adeoxoguanine induces a DNA damage response at the telomere. We looked for markers like gamma H2AX and 53p1 and the telomere red PNA fish. So this damage response is activated throughout the cell, which is indicated by activation of the ATM and CHEK2 pathways, which we also showed drives p53 expression. And importantly, when we looked at the senescence and its reliance on p53 or p16, we found that knockout of p16 in either cell line did not rescue the cell growth defect observed in the wild-type cells 4 days after induction of telomere adeoxoguanine. However, p53 knockout did, and p53 knockout rescued other phenotypes, and here I'm just showing, for example, the EDU incorporation phenotype. So to understand what's happening at the telomeres to drive senescence, we performed metaphase spread experiments, and we looked for signal-free ends and fragile telomeres. Signal-free ends are chromatid ends that lack a PNA staining, and we didn't observe really any strong induction of signal-free ends. However, we did observe significant increases in fragile telomeres, which are markers of telomere replication stress. So this data and others we have generated show that adeoxoguanine disrupts telomere replication. However, when we looked at telomere shortening by the traditional telomere southern blot, or this technique that I'm showing here, the Tesla assay, which specifically amplifies critically short telomeres, we did not see an increase in telomere shortening or critically short telomeres, showing that telomere adeoxoguanine is sufficient to drive senescence independent of telomere shortening.